Hello there. We now know from our previous lessons that the stomach is a place of storage as well as mechanical and chemical digestion. We know that the gastric juices contain hydrochloric acid, which both activates the zymogen into pepsin as well as denatures proteins. Protein unraveling is a type of mechanical digestion, and pepsin, the activated protease enzyme, is responsible for actually breaking bonds between the amino acids of the proteins. So the amino acids are the individual subunits of the protein, and pepsin can break the protein bonds between these amino acids to liberate the small subunits themselves. We also know that the gastric juices contain intrinsic factor, which will be important later for the absorption of vitamin B12. Now, the stomach, of course, accepts this bolus of food, this bite of cookie mixed with lots of enzymes and saliva, etc. And it is going to churn, mixing this bolus, right, the bite of food, with all of these different gastric juices. Okay, and so the more cookie you eat, the more your stomach is going to stretch out, and this is actually going to reflexively cause even more churning, even more mixing, more mechanical digestion, and mixing with enzymes to promote chemical digestion. Okay, so ultimately, all of this churning, all of this breakdown is going to push the chyme, right, the food mixed with gastric juices, all the way down to the pyloric region. Okay, the circular muscle, the pyloric sphincter, which is responsible for controlling the flow of chyme into the duodenum, is going to be triggered to temporarily relax. So it's always closed. And so when enough churning, when enough digestion has occurred, the pyloric sphincter relaxes a little bit, allows a little bit of chyme to flow through, and then it snaps shut once again. Essentially, this allows a little spurt of about 3 milliliters of chyme into the duodenum. Okay, so we don't want to empty the entire stomach into the small intestine all at once. That is, first of all, damaging to the small intestine, right? It can't take up that much acidic stuff. Secondly, if all of the food, right, the chyme, the bolus, all of these things go into the small intestine all at the same time, ultimately, we are not going to have enough of an opportunity to digest and absorb all of those nutrients in your cookie. So we allow just a teeny bit of chyme into the small intestine, let the small intestine process it a little bit before we let another about 3 milliliter spurt into the duodenum, the small intestine. And so let's now focus on the small intestine. This, of course, is the next organ in line in the gastrointestinal tube or the GI tract. Okay. In general, you should think about the small intestine as the major organ of both digestion and absorption. Okay. And so in order to maximize both the breakdown of molecules and taking them into our body, we need to increase the opportunities for these processes to happen. And so what I'm getting at here is that the small intestine takes every possible opportunity to increase the surface area available for digestion and absorption. First of all, the small intestine is in a straight line, as you surely are familiar with. Instead, this tube is actually 20 feet long, and it's all coiled up to fit neatly within your abdomen. Okay, so this entire thing is about 20 feet long, so greatly increasing our surface area. We will see some microscopic adaptations to increase absorption as well. Okay, the small intestine, in all its glory, is divided into 3 subdivisions or suborgans. First of all, we have the duodenum, which is what accepts the chyme in 3 milliliter spurts from the stomach. This shorter tube, right, about 10 centimeters or so, is actually retroperitoneal. And so what that means is that it is actually outside the peritoneum. It's not covered by mesentery. It doesn't have any layer of peritoneum covering it. So Here is a sagittal section looking from the right side into the abdomen. Here's the liver. Here's most of the small intestine. We can see the parietal peritoneum is this blue line right here. So all of these organs are nice and secured. They're nice and protected by this cushy serous membrane. What is not covered and protected by the serous membrane, on the other hand, is the duodenum. And so we can see it right here, posterior or retroperitoneal to this serous membrane. And if we look from the left side, we can see the stomach with the greater omentum hanging off the front. Okay, we can see lots of different parts of the small intestine sectioned through in this sagittal section. Okay, and the mesenteries, right, we can see are wrapped around these tubes, right, the different parts of the small intestine, and essentially providing a route for blood vessels, nerves, etc. Once again, the duodenum way back here is retroperitoneal, as well as the rectum, the uterus, and the urinary bladder. We'll get to there at another point. Okay, so duodenum is retroperitoneal. It is by far the shortest, but this is where all of the different digestive enzymes and buffers and all sorts of things are mixed with the food for the first time. The next section is the jejunum. And finally, the jejunum empties into the ileum. Okay, the ileum is the longest portion of the small intestine, and this is where most of your absorption is going to take place. Okay. Another function of the small intestine is actually to prevent infection, right? So as we go through the next couple slides, think about how we need to increase surface area for more nutrients to come into our body, and also we don't want anything but nutrients coming into our body. Okay, so first of all, increasing surface area. First of all, the small intestine lining isn't smooth, right? Yes, this tube is 20 feet long, so we already have a lot more surface area than we already did, but we also fold the lining of our intestine, so this mucosal membrane, into essentially swirly folds. And so you can think about like a screw or a drill bit. It has these ridges that are swirled, right? And what this does is it actually, instead of sending the food through the intestine in a straight line, it is going to swirl it around. And so this is going to mix the food together with more digestive enzymes. It is going to fling the chyme to the sides of the walls of the small intestine. Therefore, they can interact with the intestinal lining, which is covered with enzymes and able to absorb the nutrients from the chyme. So this is very effective at spiraling the food, at increasing the absorption, and increasing the digestion as well. And so these structures are called circular folds, or more technically, the plicae circularis. So if we take a section through the small intestine wall, we can of course see the muscularis externa. The serosa is out here. Submucosa houses all these blood vessels, and there are circular folds, right? Big circular folds here. Now, if we zoom in even farther, we can see that on these deep circular folds, there are little finger-like projections, and these finger-like projections are called villi, or villus for singular. We can see that in each finger-like projection, there is a capillary bed, to be continued, as well as a little green vessel. This is actually part of your lymphatic system. Okay, so that's in each one of the villi. Now, if we zoom in even farther, right, we can see that the villus is covered with simple columnar cells, right? So one cell layer thick is the boundary between the outside world, or the lumen of the GI tract, and our capillary bed, so our body itself. Okay, so these cells are called enterocytes. They can also be called brush border cells. And the reason for that is that we can see that there are little cytoplasmic extensions. So these are not cilia. They're not like creating waves or anything like in our respiratory system, but they are, go figure, greatly increasing the surface area. Now, these brush border little projections here aren't only increasing the amount of space of the cell itself, but also they're covered with enzymes. And so these enzymes are responsible for digesting. They're responsible for activating different zymogens. Right? So lots of different enzymes, which of course are constantly encountering food, which is flung to the side, excuse me, of the small intestinal lumen by those plicae circularis, or circular folds. And let's look at this in a slightly different way here. Again, circular folds are allowing for the swirling here. Each of the circular folds are covered with villi. Okay, the villi, or the columnar cells themselves, have their own little brush border. Okay, these absorptive cells, right, the enterocytes, are the barrier between the outside world and our capillary bed. Now remember that the capillaries are bringing blood from the left side of the heart, right? So it's oxygenated blood into each of these villi of the small intestine. They dump off oxygen, pick up carbon dioxide, but not just metabolic byproducts. They're also going to be picking up all of the lovely food that we just digested. Right? So the bits of cookie that we can take across these absorptive cells and into the capillary bed. And so on the venous side of the capillary bed, we drain the blood not right back to the heart. As we know from previous lessons, the blood has to make another stop first. Okay, so this blood is going to be drained up and into the liver via the hepatic portal vein. Right, those hepatocytes are going to modify it, pull out all the stuff that we have plenty of, store it away, take out anything that doesn't look quite right, get rid of it, right, break down, use red blood cells, all of these different things before allowing the blood to return back to the heart. Okay. We also have another circulatory structure, a little bit different though. This is called a lacteal. It is green, indicating that it is part of your lymphatic system. So in this lesson, we will get to learn what lacteals are actually for. All right, so the small intestine greatly increases the surface area in any way it possibly can. It's long, it has folds. Folds are covered with villi, villi are covered with microvilli, right? Lots and lots of surface area. So we want to have all this room to absorb nutrients. But what this also does is it opens us up for infection, right? If we have more opportunities for nutrients to come in, this is also allowing more opportunities for bacteria and other pathogens to come into our body as well. And so there are specialized structures called intestinal crypts, or formerly called the crypts of Lieberkühn, that are found between the villi of the small intestine, so these here, which have a major role in protection of the body, but also in replacing the lining of the intestine itself. So let's take let's take a look at a short clip from the Howard Hughes Medical Institute about these crypts. Let's see what they do in action.
There's a cross section of that sort of tower, which is known as a villus. And the villus is lined by cells on the outside that are the fully differentiated cells that play an important role in absorbing nutrients whenever you eat. Those fully differentiated cells come from the undifferentiated stem cells that reside at the bottom where that arrow is in what is known as a crypt. So if you could run the video now, we will see how this process works. So we're looking now from the inside at one of these villi, and now we're going to go through the cross-section view and look down at the crypt. Now, this is a video developed from real mouse studies in which you can turn a stem cell blue color through a genetic trick, and then you can watch every cell that's born from that stem cell will be blue, and you can watch it develop. The cells move up the crypt, they get into the lumen, they differentiate along the way. They'll get up to the top of the villus. They'll then complete their life cycle of absorbing nutrients and then undergo programmed cell death, or apoptosis. This is also happening in all of our colons right now. Very rapid rate. We're still before lunch. I think you're probably okay with that. So what happens if you have a mutation in the stem cell? there's a common mutation in colon cancer in a gene called APC that's important in cell fate. So the cells that have that mutation are now in that greenish brown color. They are going to have trouble making it through that final stage of differentiation, and they're starting to pile up. And therefore, a tumor starts to develop.
Okay, so clearly these crypts have a lot going on with them. As you just saw in the video, there are stem cells within these intestinal crypts that are continually dividing. Right? And as they divide, the cells are going to differentiate, right? So they're going to develop these little brush borders. They're going to express the brush border enzymes. Some of these cells ultimately become goblet cells because mucus is going to be very important in lubricating the intestines. And so they develop, they continue to get pushed up by the new cells in line. And ultimately, once these cells get to the top of the villus, they are shed and essentially digested, and the components are recycled for the next cellular division process. Okay, so stem cells within these crypts. Okay, one of the reasons why there may be digestive side effects of chemotherapy is that chemo, the chemicals that are targeting cancer cells, right, just like the tumor that we just saw in the video, but these chemicals are actually targeting any cells that are dividing rapidly, right? So your hair cells are dividing really rapidly. And so the chemo not only targets the tumor cells, but also anything else that's dividing. So your hair might fall out. Also, it may target these rapidly dividing stem cells as well. Now, as you can see from this image, this process or these individual cells only live for a couple days tops. And so they must be continually replaced. And if you have chemo treatment attacking the cells that are so rapidly dividing, you may not be able to replace these cells enough. All right, so that's why some individuals who are being treated for cancer may have digestive issues as well. Let's see, other cells that are in these intestinal crypts are enteroendocrine cells. And so these cells are producing a lot of hormones. We'll get into that at another time, but essentially they are detecting what you just ate, and they are going to control the digestive processes accordingly, right? So if you just had something super fatty, you're going to want a lot of bile. If you just had something with a lot of sugar, you're going to need to release insulin so that you don't have a blood sugar spike and then subsequent crash, right? So these enteroendocrine cells are detecting what you just had, right? How many cookies, what was in those cookies, so that we can release hormones to control the digestive process. Okay. There's also a lot of immune function. And that's really where I'm getting with this. The immune system has about 70% of your immune system is actually located in association with your digestive system, including these intestinal crypts. So we have specialized lymphocytes that are detecting stuff we've seen before, and therefore we can launch an immediate secondary immune response. We have specialized cells that express antibodies, right? So again, binding to stuff that we've seen before so that it's immobilized and it can't hurt us. And also there are cells called Paneth cells. And so these Paneth cells are responsible for releasing chemicals that inhibit microbial activity. So defensins and lysozyme, which we've actually seen before when we talked about the salivary immune function. Okay, so this image right here is showing us how the different parts of the small intestine are different. All right, so let's start with the duodenum because that is the first place that receives food. All right, we see a few circular folds. Really, the primary goal of the duodenum is to receive all these different digestive enzymes, to receive the chyme from the stomach, and perhaps most importantly, to increase the pH of that chyme. Okay, so as we're going to see here in a moment, the intestinal enzymes all function best around a pH of 8. Chyme is like a pH of 2. So we really need to buffer that chyme as soon as possible in order to be able to activate the enzymes and appropriately digest our food. This here is the duodenum wall, right? And we see a lot of little duodenal glands, which are only present in the duodenum. Okay. And these are going to be releasing chemicals that essentially neutralize the stomach acid, right? So the duodenum is the one that gets the acid, and so it is the one that needs to fix the acid problem. Duodenal glands are responsible for fixing that acid problem. Okay. From the duodenum, food flows into the jejunum. The jejunum has a ton of circular folds, lots and lots of villi. And so this is important because this is the area where we want the most digestion. Right, so circular folds make a lot of sense because, again, this is churning, allowing for the digestion to occur. Okay. Finally, the ileum, right? This is the longest segment of the small intestine, and this is the primary organ of absorption, right? So pulling all of those already digested things into our body. Okay. There are fewer circular folds, but what the ileum has that the jejunum and the duodenum both do not have is extra lymphatic tissue. So there are aggregated lymphoid nodules called Peyer's patches that are even more filled with white blood cells and other immune cells. Keep in mind that the ileum is the last part of the small intestine before we get to the bacteria-filled colon. And so we really want to make sure that no straggling bacteria make it up into our small intestine and wreak havoc. Right? So lots of aggregated lymphoid nodules, that is Peyer's patches, in addition to the intestinal crypts, in addition to the mucosal associated lymphatic tissue. Right? So this is above and beyond all of that. Okay, so here is just the summary. We can look at the histology to see some differences. That is not going to be applicable to your exam, but there it is, kind of cool, perhaps. The entire small intestine has villi and microvilli. Okay. You see that the number of goblet cells, that is mucus producing cells, is going to increase as you get to the end of the small intestine. And hopefully this makes sense. You are pulling water out, you are pulling stuff out. And so the chyme is becoming thicker and thicker, right? More and more concentrated. And so you want more lubrication to this soon to be solid substance than you needed, when it was just gastric juices and a little bit of food. Okay. There are Paneth cells within the intestinal crypts throughout the entire small intestine. Brunner's glands is another word for duodenal glands, and so they are only present within the duodenum. And finally, the Peyer's patches, they are only present in the ileum because the ileum is right next to the bacteria-filled colon. Okay, so let's see what happens when the chyme enters the duodenum now that we know a little bit more about the different features of the small intestine. Remember that chyme is super acidic and it's super rich in nutrients. Okay, so it's acidic. Remember, the stomach pH is very low. The duodenum wants to be minimally neutral. A little bit alkaline is also good as well. Okay, so as the acidic, nutrient-rich chyme is passed through the pyloric sphincter into the duodenum has to immediately respond to this very different set of conditions. First of all, it needs to neutralize the chyme. Okay, so the duodenal or Brunner's glands are immediately going to start secreting alkaline mucus to pull that pH back up. The intestinal crypts are going to secrete alkaline mucus. Now, again, pulling that pH back up. Okay, and in response to this acidic chyme, the pancreas is going to be triggered to release alkaline pancreatic juices as well. And so we're going to look at this in our next lesson together, but for now, just know that the hepatopancreatic sphincter is going to relax, allowing these alkaline juices to flow into the duodenum. Okay, and so all of this alkaline stuff is going to mix with the super acidic chyme and bring the pH back up. Again, we have a lot of different things throwing basic fluids at the chyme, implying that this is a really, really important process. We have a lot of backups. Okay. Also, the chyme is nutrient rich. And so there are cells within the duodenum that detect that there are a lot more nutrients than there used to be. And so essentially, the release of lots of different enzymes is going to occur as well. And so the duodenum is a major digestive organ. It houses, or it is the site of digestion of all classes of biomolecules, right? So far, what we have seen is that in the oral cavity, we digest carbs and lipids. In the stomach, we digest proteins and lipids. Finally, in the duodenum, we can digest it all, okay? So we use all classes of biomolecules using brush border enzymes and secretions from the accessory organs, okay? So we need pancreatic enzymes actually to digest everything. Okay. All right, so what about these extra accessory organ secretions? Well, the liver, as we know, makes and recycles to an extent the bile. The bile is then stored in the gallbladder, and when chyme enters into the duodenum, the hepatopancreatic sphincter relaxes, allowing pancreatic juices as well as bile into the small intestine. Now, what exactly is bile? I've alluded to this before, but bile in this circumstance acts as an emulsifier. That is, lipids are hydrophobic, right? Fat really doesn't mix well with water. And guess what your entire body is, right? It's water. So we need to break these big globs of hydrophobic things into much smaller pieces so that we actually have an opportunity to digest them. So here is a lipid droplet, big globby droplet. Again, they're hydrophobic, so these molecules clump together as much as possible so they don't have to interact with the water very much until bile comes along. And so bile is going to be mixed in with the lipids, and essentially it's going to cover the lipids with a molecule that has a hydrophobic side and a molecule and a hydrophilic side on the other side. So think about phospholipids, right? One side loves water, the other side hates water. And so these lipid molecules are going to be covered with the hydrophobic side pointing in, so it's nice and cozy, and the hydrophilic side pointing out so that these things can actually be processed within the intestines efficiently. Okay. Also, again, taking the big ones and breaking them down into little tiny droplets so that enzymes can actually come along and break the bonds between the monomers. Okay, so emulsification is not digestion. Okay, so it's just separating molecules away from each other. The lipases, the enzymes themselves, come along and they actually break the bonds between glycerol and and fatty acids, etc. So again, lipases do the digesting, but the emulsifiers, bile, actually makes it much easier and much more efficient to do the digesting. So pancreatic juices contain the actual enzymes. Okay, so remember that the pancreas is the only organ that makes enzymes to digest all 4 biomolecule categories. Okay, the pancreas produces amylase, or a variety of amylases actually. Okay, these molecules take polysaccharides, right, big clunky carbohydrates, and break them into monosaccharides, right, the individual subunits. Lipases take triglycerides, for example, and they break them into individual fatty acids. Fatty acids are the monomers of the lipids. Nucleases take DNA, they take RNA, and they're going to break them down into individual nucleotides. Okay. If all of these words seem very foreign to you, go back into the last PowerPoint notes and take a look at that biomolecule chart that I gave you. Every biomolecule category has a name for monomers and it has a name for polymers. Those are important words for you guys to know. Okay, so all of these amylases, lipases, nucleases are released by the pancreas via this hepatopancreatic sphincter into the duodenum in their active state, right? No big deal. The proteases, on the other hand, the enzymes that digest proteins, just like in the stomach, are really dangerous. We are made of mostly proteins, so we cannot start releasing proteases willy-nilly. Okay, we need to have them active only in the duodenal lumen, not before, right? We do not want them to digest our pancreas. We need our pancreas. So the pancreas releases a zymogen called trypsinogen. Remember, pepsinogen is in the peptic area, right? Like peptic ulcers, right? Those are in the stomach. Trypsinogen is released by the pancreas. And so what we're going to see here is that trypsin is inactive until it interacts with brush border cells. Now, the brush border enzymes are only present in the lining of the small intestine. And so the only way for trypsinogen to be activated into this super dangerous protease is to be in the intestinal lumen. Okay, and so trypsin is actually pretty swell. It can actually go forth and activate a lot of other zymogens as well. And so it's kind of a chain reaction here. Once one trypsinogen is activated into trypsin, we get the activation of all sorts of other enzymes as well. Okay, so just to clarify this, trypsinogen is inactive as soon as it winds up in the lumen of the duodenum. It is going to be activated by an enzyme on the brush border itself. It will become trypsin. Now, trypsin is a protease in and of itself, but also it can activate trypsinogen. It can activate other, or chymotrypsinogen and other trypsinogens as well. All right, so all of those words aren't terribly important for you to memorize, but the words trypsin and trypsinogen are very important. Remember, trypsinogen is a zymogen released by the pancreas. It is not activated until it is in the duodenum, where it is activated by brush border enzymes. Okay, so this is greatly going to amplify the digestive activity. All right, so the chyme is mixed with the bile. It is mixed with the pancreatic juices, chock full of digestive enzymes within the duodenum. Of course, the story doesn't stop there. The chyme is going to be pushed along via peristalsis and segmentation from the duodenum to the jejunum into the ileum. Okay. The entire time, we have the process of chemical digestion occurring. Okay, so as the chyme is pushed along, it's being broken down by enzymes. Remember that this process is called hydrolysis, so taking big stuff and breaking it into smaller monomers. Okay, essentially, the big idea, right, the main goal of the digestive system is to take food in its pure state. This is a hamburger, this is a cookie, and to essentially change the food into forms that can pass through those brush border epithelial cells and into the blood and lymphatic vessels underneath. And so earlier we took a look at this diagram right here, and it breaks down all of these different processes. Again, big take-home message here. We take one kind of molecule, manipulate it, break it down so that it can pass through the mucosa and into our body. Okay. Now, in the next few slides, I am going to walk you through what is digested where and where we absorb it, how we absorb it, those types of things. We can essentially look at this story from two different ways. We can focus on the oral cavity, what's digested there, which is kind of what we've been doing, but we could also say, how are carbs processed? Okay, and so the story of the carbohydrates begins in the oral cavity, pauses in the stomach, and finishes in the small intestine. Okay, so we're going to look at this similar content and adding a little bit on, but from through a different lens. Okay, so after digestion of all of these molecules, the carbs, the proteins, the lipids, these monomers must be absorbed across the mucosa. Again, I'm going to remind you where the different molecules are digested. And ultimately, the goal of this whole process is to take these monomers, right? So these here are monosaccharides of the carbohydrates. They are taken across the brush border enterocyte cells with specialized proteins into the brush border and finally allowed to go over the basal side of this membrane and into the capillaries. right? Proteins work largely the same way. Again, proteins are broken into their amino acid monomers, right? Using specialized proteins, we absorb them into the blood. Lipids, of course, are hydrophobic. They need a little bit of extra love. So the lipids are broken down into monoglycerides and fatty acids. All right, again, these are monomers. They can diffuse into the cells. They're packaged up in fancy little packages called chylomicrons, and they, because they are hydrophobic, have to be transported in the lacteals as opposed to in the blood. Again, we're getting there in just a moment. Okay, so carbohydrates. First, looking at the digestion, right? Different than absorption. Digestion is breaking it down. Absorption is taking it in. Okay, so we start out with starch. This is a polymer or polysaccharide. Amylase in our saliva, salivary amylase, breaks starch down into these disaccharides. So these are the sugar molecules, actually. And so if you've ever taken a bite out of a cracker and you chew it, it goes from being like starchy, salty to actually sweet. And the reason for that is that salivary amylase breaks starch down into sugar. This one, in fact, is table sugar, milk sugar, beer sugar. Okay. Nothing's happening in the stomach with the carbohydrates. The next time we get carbohydrate digestion, it is in the small intestine. And what we are using is pancreatic amylase. Again, the pancreas produces enzymes for all 4 biomolecule classes. I've said that enough times. You should expect to see that on an exam. Okay. Also, lots and lots of brush border enzymes. Note that these are not blue and underlined, so they are not ones that you would have to recognize, but we should recognize these. This is in the saliva. This comes from the pancreas. Okay, so now that we have broken those big clunky polysaccharides down into the individual monosaccharide monomers, what do we do to get them in the body? Well, the absorption of carbohydrates involves first creating a concentration gradient. Okay, so on this side, right, this is the side of the blood. This is where the glucose, fructose, all these monosaccharides are. So this is where your food is. This is where we want the monomers. And so the very first thing we have to do is create a concentration gradient with sodium potassium pumps, right? Pumps that we've seen many times before, essentially creating a low sodium environment inside these cells. Now, in the luminal side, right, this is where your food is, we have specialized proteins that allow sodium to flow down its concentration gradient from high, right, the salt in your food, to low. And again, this is a low concentration because we just generated this gradient over here. Okay. But in order for sodium to come into the cell, it needs to have a ticket. It needs to bring a friend. And that friend happens to be a sugar. Okay. So again, we create a concentration gradient of sodium, low sodium here, high sodium in the intestines. In order for sodium to flow with its concentration gradient, it brings with it, right, symport, glucose, galactose, whatever the specific sugar for this molecule is. Okay, so this is called secondary active transport or co-transport. And this is how we absorb carbohydrates, right? Once the glucose or galactose is across this brush border cell, it enters into the blood and it's taken up to the liver. Proteins. Proteins are not digested in the oral cavity. They are digested first in the stomach. Now remember that the stomach acid can unravel the proteins, but that's not chemical digestion. Pepsin is going to begin the chemical digestion in the stomach. Trypsin and the molecules that it activates, right? Chymotrypsin, carboxypeptidase, lots of different things. So these pancreatic proteases are going to work on the digestion of proteins in the small intestine, right? The pancreas, again, is an accessory organ. It never actually touches food, but we absolutely need the enzymes from the pancreas in the small intestine to complete digestion. Okay. There are also a lot of brush border enzymes that contribute to this process as well. Again, the ones that are most important, trypsin and pepsin. Nothing's happening in the mouth. Absorption. Now that we have taken those big proteins and we've broken them down into the individual subunits called amino acids, we do pretty much the same thing we did for the carbs. We generate a concentration gradient. Blood's over here, right? Pump sodium into the blood, leaving low sodium here. Now there's higher sodium on this side than there is over here. In order for sodium to flow with its concentration gradient, it needs to bring a friend. This friend is an amino acid for this particular protein. And so amino acids are then taken into the cell and then pushed into the blood. Okay, so same type of process, secondary active transport. Now, there are 20 different types of amino acids. We as humans can generate 11 or 12 of them. And so the rest of them have to come from our diet. So we absolutely need this process to work perfectly. Finally, the lipids. We digest lipids pretty much all the way down. Okay, so starting in the oral cavity, we release lingual lipase to start breaking bonds within the lipids. In the stomach, we release gastric lipases to continue this process. The pancreas also releases its own lipases into the small intestine to continue lipid digestion within the small intestine. Now, lipids are different because they're hydrophobic, and so it takes a long time. They really don't like to be in water like everything else does, and so we actually use bile salts as an emulsifier to facilitate the process of digestion. But again, this is not digesting the fat, right? It's not digesting it, it's just making it easier for pancreatic lipases, et cetera, to do the digesting. Okay, so how is this absorbed? Okay, and how is it transported? Because again, it doesn't like water. So how do we deal with this? Well, by the time you get to the small intestine, we have these big globs of lipids. Okay, bile is released and emulsifies these big globs of fatty acids down into these smaller droplets. Okay, digestion occurs. Oh, sorry. Digestion is going to occur with all those lipases, cleaving the bonds between fatty acids, etc. Okay, so at the end of this emulsification and digestion process, what we're left with are little droplets of lipids that are surrounded by specialized molecules that love water on one side, hate water on the other side. Now, these lipids can actually just pass through our membranes, right? Remember, our membrane is mostly made up of lipid, like the phospholipids. And so these things don't need any specialized proteins or anything. They can just slip right over the membrane. Okay. And once they are inside our brush border cells, they are repackaged by the Golgi apparatus, so a specialized organelle that is responsible for sorting, processing, shipping away different types of things. In this case, the Golgi apparatus is going to repackage those digested lipids into structures called chylomicrons. Now, these chylomicrons, again, are not very happy being within water. And so we can't just dump them into the blood and everything's okay. Instead, these chylomicrons are actually sucked into the lacteals. Remember, the lacteals are lymphatic vessels. They have big gaping holes in between their cells. Okay, and so they suck up these chylomicrons, right, chock full of lipids, and essentially these little droplets are milked along, right? We know that there's no heart pump, right? It just depends on the movement of the intestines, the movement of the skeletal muscles to actually squeeze all of these fats up to the liver for processing. Okay, so the reason why these vessels are called lacteals is because they're super white. It looks like milk, right? Lactation, milk, right? The root is connected here. And so the lacteals are pretty much transporting fat, right? And that's what they do. And so it looks like milk being transported, but in fact, it's just hydrophobic molecules. Okay. Ultimately, these chylomicrons are actually delivered to the liver. Go figure, the liver is going to process the cholesterol and essentially it packages it into these little droplets that are okay with being in water, right? We talked about these in the very beginning of class. Lipids that are transported by HDL proteins, right? So high density lipoproteins. So these proteins are hydrophilic on the outside, hydrophobic on the inside, and these HDL molecules essentially take lipids, right? And they're more or less controlling how many lipids are out in the blood versus how many lipids are stored within the liver. We also have LDL. Remember, this is the bad cholesterol. Essentially, this is the transport molecule that is going to take lipids away from the liver and possibly clump together within your arteries. Okay. Now, if you have a certain genetic condition, LDL is a much more commonly made molecule by your liver. And so you're going to have more of the bad cholesterol, even if you have a really healthy diet and exercise. Okay. Okay, so final topic in the small intestine before we move into the large intestine. The ileum is where the overwhelming majority of nutrient absorption is going to take place. Once the chyme passes out of the ileum into the colon or the large intestine, there is no more nutrient absorption that's going to occur. Okay, immediately we mix the chyme with bacteria, right? This is called our microbiome, our gut flora. So lots of bacteria, and we want them there. And again, it is really important for the ileum to have this extra layer of protection because there is a little valve that opens up, lets chyme go, and then closes off again. So we really don't want bacteria to sneak up into our ileum as we're letting chyme go into our large intestine. So that's all for this clip. One more for this particular lesson. Thanks.